Okay. Is it showing up on Facebook? It might be double posted. Are we on? Okay. And it looks like we're starting to have attendees come in, which is great. For those who are logging on right now, this is Popping the Science Bubble, hosted by the Berkeley Public Library tonight. And we're just going to take a minute as people log on. Just a note for folks, we are recording the presentation tonight. So the presentation appears on our YouTube page, or the Popping the Science Bubbles YouTube page. So this presentation is being recorded. The presentation is also streaming live on Facebook, or should be. Good test to make sure that works.
Yeah, I don't think I can click on live video, but I'm not sure if it's just me.
Oh, okay. Let me see if I can deal with that. All right. Okay. Well, I'm going to take a moment. I'll just start getting, I'll start with our introductions here, and then I can attend to our Facebook video. Thank you so much, folks, for logging in tonight. Welcome to Popping the Science Bubble. This is a monthly seminar series that aims to share new research findings from grad students and postdocs at UC Berkeley with the general public and create constructive discussion about a variety of science topics. We have 2 speakers at each seminar who will talk about their current research or a topic that they find really interesting. The organizers of this program are 3 graduate students at UC Berkeley, Jenna, Madison, and Oksana. If you're interested in checking out past seminars, you can visit their website and check out their Twitter or Facebook, or sign up for the listserv to keep in the loop for the upcoming seminars. And I really recommend signing up for that listserv. The Science Bubble just does a great job telling details about the talks and really talking about the topics. And there's just so many interesting and diverse topics each month. It's cool to be kept in the loop. Thank you so much for logging in tonight, and I'm going to hand this over to the Science Bubble team to introduce tonight's speakers.
Thank you, Kelsey. Yeah. Hi, everyone. Good evening. Welcome to the March 2022 seminar. Tonight we'll be having 2 talks from 2 wonderful postdocs at the UC Berkeley campus. What I want to let you guys know before the seminar starts is that we're happy to take questions as we go. So please feel free to put in the Q&A and chat any questions that you have. And then Jenna, Oksana, and I, the organizers, will be monitoring those and making sure that we can moderate any questions as we go. And after both talks, we will also be holding a few minutes of extra Q&A with each speaker. So please feel free to participate as you are curious or interested. We are obviously happy to have you guys ask as many questions as you'd like. So our first speaker tonight is Guy Neer from the Astronomy department here at UC Berkeley. He studied physics and astrophysics in Israel, where during his PhD, he worked on building an observatory in the Israeli desert. and his hobbies include science, real and fiction, and photography, both of the night sky, but of various other subjects as well. He's currently a postdoc at the Astronomy department, and we're really excited to have him here to talk to us about potential for life on other planets. So please take it away, Guy.
Thanks. Let me share my screen. Hold on. Okay. Right. So life on other planets. This is a topic that has always been interesting to me, particularly. 20 years ago when I was a kid, this stuff was really the realm of science fiction. Nobody really knew how to say anything scientific about the prospects of life on other planets. People would have sort of guesses about how many planets are out there, how many of them have life on them, but nobody really could give a really good answer to this stuff. And we'll see that in the last 2 decades, there's been a lot of progress, and the stuff that used to be science fiction is now becoming really science. So there's 2 parts to this question of life on other planets. First of all, how does life start? So we know only a few pieces of the puzzle. We don't really understand how life began. Some people have done some experiments where you take abiotic materials, some chemicals that people think used to exist on Earth when it was very young, and put them in like the soup of water and all these different chemicals, and shoot lightning electrical charges into that slush. And some experiments you can see that there are organic molecules that start to form from this process after a long time. One of them, for example, is Miller's experiment. I hope you guys can see my cursor. So Miller's experiment is one example of such a classical experiment where they can actually build some of the building blocks of life from unorganic objects, molecules. Another thing that people like to bring up in this context of how life began is Conway's game of life. Let me see if I can show it. So here's an example. There are black and white pixels, and there are very, very simple rules in this game. If a black pixel is surrounded by a certain number of white pixels, it dies. If it's surrounded by too few white pixels or too many black pixels, it dies. But if it has only 3 or 4 neighbors that are black, then it becomes it can multiply, or it can generate another black pixel, or it can survive. Anyway, it's a very simple set of rules for each pixel to survive to the next turn. But even with these very, very simple rules, I'll show you how it looks when you have these specific structures that somehow formed. And you can see that even with these very, very simple rules that each pixel knows only about the few pixels around it, you can already start to have these scenarios where things move around and multiply. And this thing, for example, shoots out these little pellets that keep living indefinitely. So it's kind of a philosophical example of how you can have very, very simple rules and still produce very complex behavior. So the analogy in this case would be that life, even with very simple chemical rules that control how chemical reactions happen, if you put them together in the right order, it just spontaneously will start to create life. So this is kind of like the philosophical way people are looking at it. Some people are doing experiments. There are a lot of problems with this. For example, our bodies, all living creatures are made out of proteins, basically. And the proteins are made by transcribing DNA, information on our DNA. But to produce DNA, you need proteins. So it's kind of a chicken and egg problem, because you don't know what came first. If it was the DNA, then what created the DNA without the proteins? And if it was proteins, then where were the blueprints for those proteins? So there's a bunch of different theories about how this came to be. So some people say that very simple RNA, which is the messenger between the DNA and the proteins, was actually the first thing that happened, that was created, and it would self-replicate. There's a lot of different theories, and the bottom line is that we really don't know how life began. We have only some ideas. There's also another thing that a lot of times gets thrown around, which is panspermia. It just means that life landed here from some asteroid that hit the Earth or something, which in my mind doesn't really help you solve the problem. It just means that life started on another planet and got here somehow. So problem of how life began is still unsolved today. But there are good answers if you ask the question, when did life start? So we can find very, very, very small fossils embedded inside crystals that we can actually date the crystals based on nuclear radiation and nuclear isotopes that decay over millions and billions of years. And the more people find these microfossils, the more they figure out that actually life was more ancient than we thought. It used to be that people talked about life starting 2.5 billion years ago and then 3 billion years ago. And it starts to go back and you find more ancient fossils, which are harder to really tell if these fossils are living creatures or just molecules that look like living creatures, microscopic life forms. But right now it seems as though life started very, very early in the history of the Earth. So if the Earth is 4.6 billion years old, then some evidence suggests that life started 4 billion years ago. So it's actually a very short time after the Earth was formed and cools down enough to have liquid water. It almost immediately started life. So what it means for me, in my mind, that tells us that actually everywhere, if Earth immediately started to have life on it when it was formed, or a very short time after it was formed, that means that everywhere in the universe where you have a planet similar to Earth with the right conditions, you would probably have life very shortly after that planet cools down. So
hi, Guy. We have a question from the audience.
Yes.
They asked, have you ever read Richard Powers' book, Bewildered, which features an astrobiologist?
No, I haven't read that one.
Okay. Maybe if the audience member wants to add a little bit, they were curious about how accurate his descriptions are of extremophiles and alternate universes. I don't know if you have any thoughts on those two topics.
Well, I don't know specifically what the specific author is talking about, but yeah, extremophiles. This maybe goes back to this picture of these vents that are under the oceans and spew out chemicals and heat, and people think that life may have started under these conditions. And yeah, there are bacteria that live in very harsh conditions that can survive radiation and heat and all sorts of conditions that for us seem very alien. So yeah, it's very likely that there are bizarre places in the universe where conditions are very different than what we know about. So we kind of expect to find life where there's water and where the temperature is not too hot and not too cold. But of course, there may be life forms in very extreme conditions that we've never thought about. Mostly I'm going to focus on the stuff that we do know about, because this is the only thing that we can really say with some amount of scientific certainty that we know that it can exist. So I don't want to speculate too much about creatures that live in extreme environments because we just don't know. Yeah, so here's a timeline of life on Earth. So basically this red part is where the Earth forms. Somewhere in this orange part is kind of like where life began. And we're thinking maybe it started really quickly after the Earth cooled. And then for a very, very long time, all this yellow bar here, it was simple organisms. So either single cell organisms, at some point start to become multi-cell organisms. At some point, about 2.5 billion years ago, some bacteria started producing oxygen, which was a catastrophe because except for them, everybody else, all the other bacteria couldn't metabolize oxygen, was very poisonous to them. So lots of different bacteria actually died away because of this oxygen, but then the other forms of life that could use oxygen were much more efficient. And as you know, today, most forms of life actually breathe oxygen. And then at some point, about 500 million years ago, life started to become more complex. We started to see the familiar shapes of creatures that we know today. Here in the green part, you have dinosaurs. And here in this little white sliver of a pixel at the end of this bar, I've expanded this last 7 million years, just so we can see better. So out of this 7 million years, you can see that humans only occupy a small part of even this bar. And even then, agriculture, 10,000 years, and advanced technology all squished up in this last sliver of the bottom bar. So you can kind of see the difference between how long the Earth had only bacterial life or simple life, and what small, tiny fraction of the Earth's history there's been intelligent culture and technology. So it's actually a really, really small part of our history. So that also tells us that if we find life on another planet, most probabilities it's going to be microbial life. It's not just by the statistics we have of our single planet that we can extrapolate, which is always a dangerous business. But most of the life we're going to find is probably going to be simple, very simple life forms. The other question that we should ask in this context is the Earth special? We're at the right distance from the sun. We have the right kind of atmosphere that you can produce oxygen, and you can use carbon to build all these complex molecules. You have water, which is a great solvent, and it's very useful for starting life, and it's very useful for living creatures. Some people say magnetic field is required. So we don't even know exactly how common are planetary systems, right? We need to know if all the stars that we see in the sky, and we know that our sun is just a star like all the other stars in the galaxy, of which there are hundreds of billions of stars, but how many of those have actually planets like the Earth circling around them? How many of them are in the right place in terms of distance? How many of them have water on them? And all these conditions. So one thing that you might think is that maybe human beings are exceptionally lucky that we are here to live on this great planet that's right at the right spot. But to that, I would say that there's something called the entropic principle, which just says that if we had not been living creatures on this very comfortable planet, then we obviously wouldn't be having this conversation and all of astronomy and all of the research into the origins of life. So it's kind of the only way that we can have these questions, these philosophical questions or these scientific questions, is by actually already having been lucky enough to be on this planet. So it's kind of doesn't require a lot of luck. It just makes sense that if the conditions weren't right, we just wouldn't be here. So we'll come back to the entropic principle towards the end. So any questions about this, about life on Earth so far? All right. Okay, so where can we find planets in our solar system? Where can we find life? Maybe in our own solar system. So the checklist is not very long. Mercury is very, very hot and has no atmosphere. Venus has a hostile atmosphere. It doesn't have any water on it. Although recently, if you want to look for it, some initial signals of life of molecules produced by life that was found in the upper atmosphere of Venus. So maybe it's very preliminary. Mars probably used to have liquid water, but now its atmosphere is very thin, so probably doesn't have any life on it right now. Jupiter, Saturn, Uranus, Neptune are gas giants. They have no apparent surface that you can stand on and produce life, so probably not on the gas giants, but maybe on one of their moons. So there are a few moons that are really interesting in the solar system that we know may have life. So Titan, which is a moon of Saturn, is very large, and it has a methane atmosphere instead of nitrogen and oxygen like we have. It's extremely cold, but the methane in its atmosphere actually becomes liquid. So it's conceivable that the methane actually takes the role of water, and there are living creatures in the oceans of methane that are on Titan. So this is one active topic of research. Europa, which is a moon of Jupiter, and Enceladus, which is another moon of Saturn, are actually covered by ice. And under the ice, there's an ocean of liquid water. So it's possible that in those oceans there are life. Here in this picture, for example, you can see Enceladus with cracks in the ice that are spewing out water vapor into space. And this is from the Cassini spacecraft that flew around Saturn. And they took these pictures of plumes of water. These are basically water volcanoes erupting from under the ice. There's a bunch of other moons that also have these, may have under ice oceans, all which are potential for life. So all of these places are interesting when you want to search for life, but It's only a handful of places that you can go, and there are NASA missions being planned to go to these places to actually look for life. But if you kind of want to expand your search, you want to look at planets outside our solar system, what we call exoplanets. So how can we find exoplanets? First thing you have to know is you can't see them directly. So for example, this picture of this star with a planet transiting in front of it is not what you see when you look at stars outside our solar system. This is a picture of our sun and Venus transiting. So if you look at a star that's outside our solar system, usually you just see a point of light. You don't see the planet. You can't make out the shape of the star. It's way, way too far for all the optics that we have today. But what you can see is different ways of detecting these planets. For example, if this planet is transiting in front of the star, you can see it kind of blacks out a small part of the surface of the star. So the amount of light you'll see from the star goes down. So here you see lots of measurements of the star, of the star's brightness. And then for maybe a couple of hours, it will drop and it will go back up. So when it drops by this very tiny percent, because the planet is usually much smaller than the star, then you can say, okay, so this might be a planet. And this is actually the best way we know of to find planets. And there are a few thousand planets detected with this method. Another method is actually to measure the motion of the star as the planet goes around it. It kind of also swings the star back and forth. And because it's swinging towards us and away from us periodically, then you can measure how the different wavelengths of light change because of the motion of the star. There are other ways using microlensing and astrometric shifts. And in some cases, you can actually see the planet directly with very, very specialized optics, but this is very rare cases. So let's just take a tiny glimpse at how this history looks. So end of the 80s, start of the 90s, people started to find planets. It was very hard at the beginning. They would see this one thing that they thought maybe it's a planet around another star. Then they found another one. They found a couple more, and kind of went very slowly. In the 2000s, people actually detect a few exoplanets directly using these specialized optics. And then in 2009, NASA sent up the Kepler Space Telescope. And the Kepler actually had very, very sensitive optics, and it could discover these very, very small decreases in the amount of light, and it found thousands of planets. And based on the information we have from Kepler and from current missions, we actually can say that right now the number of planets in the galaxy is of the order of the number of stars in the galaxy. So that means on average, every star has at least one planet. So this is the Kepler mission. This is a couple of examples. I won't go into them too much, but you can kind of see these star systems with their planets. And we can't really know how the planets look. We can estimate their size. So we don't really know if this thing has a solid surface or covered with water, or there are some models. But basically, all we know is that the dip in the amount of light you get from the star. So that tells you something about the size of the planets, and not much more than that. But you can kind of see there are all these different systems. Some of them are very exotic. They have lots of planets. Some systems, the planets there's an example of a planet where you have multiple planets around one small red star, and all of them in an orbit that's closer to their star than what Mercury is close to our sun. So you can imagine if you're sitting on one of these planets and in your sky, you could see these other planets moving around your sun the same way we see the moon orbiting around the Earth. So it's going to be pretty interesting to live on such a planet. But the upshot of all of this is there are billions of planets in the galaxy. There's no shortage of places, and lots of them are actually in the habitable zone. So in the right place in the right distance from their sun to have liquid water, and lots of them are the size of the Earth or similar to the size of the Earth. So probably lots of them are rocky planets with liquid water on them. So I also want to mention TESS, which is another experiment that was launched in 2018, which had a wider field of view, and it was specialized in looking for planets that are nearby our solar system, just because it's easier to then get better information about their atmosphere with the James Webb's telescope, for example, which has now been launched finally at the end of last year after many, many delays. And the most important thing for me about the James Webb Space Telescope is that it can actually measure different wavelengths of light from the planets, not just from the star, but actually to tell apart what comes from the actual planet. And if you know which optical signatures come from these atmospheres of these planets, you can tell if these planets have different materials, for example, molecular oxygen. So oxygen, like we said, actually was produced on Earth by living creatures, by microorganisms that actually use the sun's light to do photosynthesis and produce all this oxygen, the same way plants are doing it right now. And actually, it's very hard to find an atmosphere that has all this molecular oxygen in combination with other molecules without the presence of living creatures. It doesn't happen spontaneously, but according to the laws of chemistry that we know. So if we find these signatures of oxygen, be really indicative of life, at least microbial life. So James Webb is supposed to be able to actually look at these planets that TESS is finding and Kepler has found and tell us if there are biosignatures on these planets. So it's actually a really exciting time for search for life.
Hey, Guy.
Yes.
Sorry, just a quick question. That's super cool, but how long does it take to like scan all of the potentially habitable or, you know, planets that you would think might potentially have biosignatures?
Right. Yeah, that's a good question. The James Webb Space Telescope is really one of the most advanced tools that we have in the sky right now. It's still not producing data. It should be in a few months. And when it starts producing data, everybody's going to want to use it. So you have a very limited amount of time on this instrument. So what people are doing is they're actually trying to model the planets that we do know and use the information we do have on them to prioritize which planets are most promising. When you have a planet in the habitable zone, in the right size, with all the correct indications for something that might host life, then we start by looking at those. Eventually, it could look at hundreds of planets. So it's very hard to look at the atmosphere of a planet because the planet is very dim compared to the star. So you kind of have to point the telescope for a very long time. I think people are doing this right now with the Hubble Space Telescope, but Hubble is much smaller. So just in terms of the amount of light you can collect with this, the giant mirror on the JWST is going to be very useful. Also, it works in infrared, so it's better to see the planet. The contrast with the star is smaller in infrared. Any other questions about this? Okay, so
thank you.
Bottom line of this part of the talk is that life probably forms as soon as conditions are ripe. Planets that are similar to Earth are pretty common in our galaxy. And we can now have the technology that's actually up in space that potentially can detect biosignatures, at least for planets that are nearby the solar system. So I think it's quite likely we're going to find signs of microbial life on other planets in our own lifetime. So probably in the next few years. I don't want to say a number of years, but probably not very far from us now. But most people, what they want to know about is intelligent life, right? This is actually a lot more interesting question than this question about microbial life. And here we have to be a little bit more speculative because, again, we only know of one planet that has evolved intelligent life, and even that we're not sure of. So there are some considerations. Some people are saying that this doesn't make sense. There's something called the Fermi Paradox, which basically says, if other planets are common and they have life on them, and some of them have intelligent life, why don't we see them in the galaxy? I mean, they should be everywhere, right? And the crucial part of it, I think, is if you look at a few things. For starters, the current rate of technological advancement, right? It took us only 10,000 years ago, we started building agriculture and cities and stuff like that. And the distance between each revolution was very, very long time. And now today it's like every tens of years, we have some revolution that changes all our technology and everything changes completely. 50 years ago, we made it to the moon. Now every 10 years, you have something like the internet showing up. So it's kind of hard to tell where we're going to be in 1,000 years or in 10,000 years of technological development. We're probably going to be much, much further along in terms of engineering and our ability to send people to distant places in space. So the question is, how much time would it take a civilization like our own, if you make it 1,000 or 10,000 years more advanced, how long will it take us to travel and colonize another star, or at least a planet around another star. It could take, in terms of light years, these stars are tens of light years from us or hundreds of light years from us. And assuming we can travel, even at 1% of the speed of light at some point, it will take us tens of thousands of years to reach these stars and colonize them. And then tens of thousands of years to build up on those planets and from them to colonize other planets. But the total time, because each planet can send multiple spaceships to colonize other planets, in principle, it can take you, millions or tens of millions or hundreds of millions of years, but eventually you will colonize all the different planets of the galaxy that are worth colonizing. So it kind of makes sense that if other civilizations existed before ours, and Earth is 4.5 billion years old, but there are stars in the galaxy that are 10 billion years old. So probably there are other places where life started that were billions of years before us. So if they had all this time to develop intelligence, to develop space flight, to start colonizing the planets in the galaxy, they should probably be here already. So it kind of doesn't make sense that we don't see anybody else unless somehow we're special and we're the only ones. So there's a bunch of different explanations. Some people say intelligent life at some point just tell themselves that it's too dangerous to meet other civilizations for different reasons and they just hide. Maybe we're just making stuff up right now, and when we actually figure it out, it will turn out that traveling to another star is so expensive, it's so complicated that just nobody's going to bother to do this for millions of years in the future. Nobody's going to bother. Maybe they are colonizing, but just on the other side of the galaxy, so we kind of don't see them. All these explanations, in my mind, are kind of okay explanations if you apply them to one or two civilizations, but if you think about all the different civilizations that might be out there, it kind of doesn't make sense that not even one of them would want to colonize the entire galaxy, would feel it's important enough. Maybe we're just lucky. Maybe we happen to be the first one. So this is kind of the optimistic approach. And then there's a pessimistic approach that says, well, everybody like us reached this age of nuclear physics, and then they destroy themselves very quickly. Just by having a giant war and throw themselves back in the Stone Age. And this is just the way it goes for all the species in the galaxy. This kind of like human nature or intelligent nature. And that's why you don't see anybody else, because nobody's managed to pass through this phase of nuclear war. So for me, I think my personal opinion on this is that it's somewhere in the middle between these two explanations. So probably a lot of civilization end up destroying themselves. Probably intelligent life is not that common, because you saw on the time scale that it took billions of years until intelligence came up from like simple life. So maybe this thing is not so common. Maybe only a handful of planets in this galaxy have intelligent life on them, and maybe some of them already managed to destroy themselves. Maybe some haven't. And maybe we're just lucky, because if we weren't lucky, then it would be the entropic principle again. And just if we were the second species not to destroy ourselves, then somebody else would have already visited us and gave us all the answers, and we wouldn't be here to do this talk, and we wouldn't have astronomers because all the information would already been given from the aliens. So in my mind, it kind of means that we still have a chance not to destroy ourselves and be the first ones. But of course, we still have the chance to also do destroy ourselves, and that puts us in an even more unique position to not do that if we really are the first ones in the galaxy. So that's my conclusion. Thank you.
Great, thank you so much, Guy. That was a super cool talk. We're happy to answer any questions in the Q&A and chat as they roll in, but I might start off with a couple. The first one is pretty naive. So obviously, like water and or other kind of nucleating chemicals seem like they'd be a good sign of life, but also, right, like one big sign of our life is how much junk is around our planet or in our atmosphere that like just isn't natural at all, or like isn't organic. Is there like any program that kind of scans our galaxy for things that like are considered astronomically unnatural?
Yeah, so most of the stuff you're talking about is not easy to detect. So you could say that, for example, all the carbon dioxide that we're throwing in the atmosphere, that's kind of the most obvious thing that humans, that industrial humans have been doing to the atmosphere, right? But carbon dioxide can also be emitted by volcanoes and stuff like that. So just seeing the CO2 doesn't tell you that you're looking at life. Other junk that we're producing, like plastic in the ocean or satellite debris in space, it's not going to be visible with the technology that we have. Some people are looking for more extreme examples. So for one of the ideas is called a Dyson sphere. So basically what it says is that a super advanced civilization would need so much power, so it will build basically giant solar panels around the sun. And then people are saying, well, if you see a star that's being dimmed, with a planet that's dimming it, but very extreme dimming, and it's kind of not periodic, it's got a weird shape or something, then maybe that's a signal of a super advanced intelligent civilization. I've seen some papers about that, but most of the time you can explain it with simpler things. So for example, if there's a planet going close to the star and it's breaking up into like a cloud of rubble, basically, that rubble will look the same as this Dyson sphere. So you wouldn't be able to see it. Other people looking, because this sphere would actually convert all the star energy into infrared. So people are saying that if you see a star that has this weird infrared excess, then maybe that's a telltale sign of but these are all hypotheticals. I mean, nobody guarantees that any of these Dyson spheres actually exist, right? So it's just an idea verging on science fiction. But mostly people are looking, when you talk about industrial society, people are looking for radio signatures. That's like the obvious thing, because that's what we're spewing out that's most easy to see. But even that, if our planet, for example, if people were looking at our television and radio communications from space, let's forget the fact that it's only been a few tens of years that we've been broadcasting, say 100 years. So we're only visible in the 100 light years around the Earth. So it's not a lot. The whole galaxy is tens of thousands of light years. So if we live on for many thousands of years in the future, then our radio signals would reach the edges of the galaxy, but they would probably be very, very weak and we wouldn't be able to detect them. So an alien civilization that's like on the other side of the galaxy, it's not in our solar neighborhood, we won't be able to see it just by radio transmissions similar to like TV and stuff like that.
Cool. Thank you. That was very thorough. I appreciate it. And then also, I was just wondering, I guess, like in your day-to-day research, what are you looking at? Like, are you looking at planet signatures? Do you look at like, what might give possibility for life?
Right. Actually, I'm not working on exoplanets right now. My work is more about other astrophysical objects, mostly white dwarfs, black holes. But that's, I think, a topic for another presentation.
Gotcha. Cool. Thank you.
This life on other planets is more of a hobby for me.
Well, it's not too shabby of a hobby. It seems quite cool.
No, but I have to say that in Berkeley, we do have a large SETI lab. So people are really doing their research on looking for extraterrestrial intelligence. Like I said, radio signals mostly, but other stuff. So there are people that really have, their job is actually to look for these things.
Cool. And maybe before we move to Shivani's talk, there aren't other questions coming through, but kind of related to Sarah's previous question about extremophiles, is there any thought to like what kind of signatures are out there that wouldn't be consistent with life like ours, or like what kind of signatures are just we not even thinking of?
Right. Yeah. So as I said, there is some thought being given to methane-based biology, because just because we have Titan and it's very close and we can actually see if it's there. But in terms of looking for biosignatures, it's really hard to start imagining how life would be with a different chemistry or different sorts of molecules that you can use. And because that's much harder and it's not guaranteed to be to even exist, then it's kind of less tempting for scientists to work on that. I'm sure people are working on this, and in some obscure journals, there are probably people publishing papers on this with different models, but it's all very speculative. And my approach is that you really should work with what you know is really exists, and not and put most of your resources on trying to find what we actually know could be out there. And if you want to start imagining other things, then yeah, of course you can do the research, but it's going to be a long shot, and it's probably not worth your time looking for that before you even found stuff that you do know about. It's kind of a problem of priorities mostly.
Gotcha. Great. Thank you so much, Guy.
Sure.
I think Guy's going to stick around through the next talk. So if anyone does have any questions that come up as our next speaker is talking, you are free to put them in the chat where they can be answered. But otherwise, Oksana will introduce our next speaker.
Our second speaker tonight is Shivani, who grew up in India and did her PhD in Mumbai. There, she studied algae and found how photosynthesis is regulated by key growth regulators in algal cells. Since then, she has been interested in understanding how algal cells fix carbon by photosynthesis and convert it to oil. We are very excited to learn more about this topic. Take it away, Shivani.
Thank you. Let's get back. So I'll be talking about Earth, and let's talk about life on Earth and specifically algae. So after that great talk, I'm glad to get back to life on Earth and organisms that fix carbon dioxide and produce oxygen. So here is a picture of that I took of the algae that I work on, which I'll be coming to eventually, but they actually turn from a green color to an orange color while simultaneously producing oil. So this is basically what my talk is about. Okay, so algae as small oil factories. Recently, we have noticed that there is an upsurge in gas prices, especially in the Bay Area. This is an image of the gas price in the Mission San Francisco area where I live. And as it is clear in the past few days, they are at a record time high. This gas or gasoline is petroleum. It's a category of fossil fuels, which are used as energy. The consumption of fossil fuels in the world is actually very high, as can be seen over here. And especially oil consumption is one of the highest in countries such as United States and China, and even in the country that I come from, which is India, suggesting that there is a huge usage of oil as a form of energy. But one has to remember that fossil fuels are a non-renewable sources of energy, in the sense that once used, once depleted, they cannot be replenished. Now, this particular form of, this increase in gas prices is definitely impacting the daily lives of people. And this is important because gas, which is abundantly used, is producing which releases CO2 upon combustion, produces climate change and a host of other factors. Now, talking about climate change and CO2 and algae. So what is up with climate change and CO2, and where do algae fit in? And to answer these questions, when we are talking about climate change and the increase in CO2, one must understand that the global carbon budget, as in how much is being produced from the sources and how much is being consumed by the sinks. So as cannot be more clearer from this picture, whopping 89% of the CO2 is released by the combustion of these non-renewable fossil fuels, while terrestrial decay and a process called respiration of plants and other organisms produces 11% of the CO2, which is part of the carbon cycle. More importantly, the major sinks of carbon dioxide are at the atmosphere, which can tolerate up to 48% of the CO2, which is 18.6 gigatons, while plants and marine life can each contribute to around 29% and 26% respectively. Now, what is marine life majorly constitutive of? And the major life that is present in marine, which constitutes marine life, is algae. And you can also think of algae as renewable sources of energy. So more importantly, just like land, the ocean also contributes to absorbing CO2, as you can see. And as you can see that it is a major sink. Now, considering both these sources and sinks, you can see that there is, if you consider all the calculations, you can see that there is a 3% deficit, in the sense that there is an excess of 3% of CO2 that is being produced by mostly because of anthropogenic activities. Now, more importantly, what algae and other marine organisms and terrestrial organisms do are they produce oxygen. Now, primarily the fossil fuels that are being burned are also a biomass obtained from the ocean floors after millions of years of fossilization of algae. However, that biomass, once lost, is lost forever. But now, but the algae in the oceans can be considered as a renewable source of energy, unlike the fossil fuels. Now, coming to what exactly are algae? Now, algae are an extremely diverse set of photosynthetic organisms. They vary in their shapes as well as sizes. For example, the smallest algae is called Osteococcus, and this is around as tiny as 0.8 micron, while the largest algae is called a giant kelp, which is around 150 meters long. Now, there is a very varied size between these two. However, both of them are algae. So what makes an algae? So algae are primarily photosynthetic in the sense that they take up a lot of carbon dioxide and release CO2 and fix this carbon dioxide using sunlight into sugars and other energy forms. And also they are mostly eukaryotes in the sense that they have a nucleus. But there are also cyanobacteria, which are called blue-green algae, but most of the algae are eukaryotes. Now, they come in a variety of different shapes and beautiful structures, as you can see over here. And therefore, they constitute a diverse form of living photosynthetic organisms. Coming back to their ability of fixing carbon dioxide from the atmosphere, one has to, we do have to grapple with the reality in the coming years that there is an increasing amount of CO2 within the atmosphere, which is where you can see that the predictions are that it will keep on increasing in the upcoming years. Now, so it is important to understand the fundamental biology of these photosynthetic organisms, and especially those which constitute around 26% of the sink within the atmosphere. So these organisms, which are algae, they're photosynthetic, as I said, they convert atmospheric CO2 into oxygen and sugars. And also they convert this carbon dioxide into oil, and also produce a huge amount of biomass, which can be further converted into oil. So what are these specific conditions and what is the biology is what I'll be talking about in the future. And what is oil? What is the fundamental structure of oil? What does it constitute? So oil is also known as triacyl glycerides. Triacyl glycerides suggest that they have a carbon chain with 3 carbon atoms that are further linked to long chain fatty acids, as you can see over here, depicted over here. And these triacyl glycerides are called oil. Now, from now on, I shall be referring these triacyl glycerides as tags or oils, because oils are made of tags. And for example, the cooking oil that we use. So cooking oil is also made of tags. They are good and delicious. But more importantly, tags can also be converted into highly dense form of fuel, which is used in transportation called jet fuel. Now, therefore, they have huge industrial applications. And because they are a renewable source of energy, they have the potential of becoming carbon neutral, and they do not produce any carbon deficit. Well, in principle, but at an industrial scale, the situations become more complicated. So what do we do in the lab, and what are some other algae capable of doing? So what I said was, when you have algae and you provide some sugars or carbon dioxide, or you provide stress or some other environmental change, such as increased salinity or temperature or increased light, under a variety of conditions, these algae further convert these carbon dioxide into oil or biofuel. And here I'm showing an example that has been studied in the lab, where of an algae called Chromochlorus ofingensis, which is photosynthetic under normal conditions. But when you provide certain conditions such as stress or nitrogen or a starvation of a nutrient or high amount of carbon dioxide, they convert into, they produce large amounts of tags, which are represented by the yellow color, as you can visualize. Are really interesting. Now, what I'll be discussing in the next slide is about my work and what we study in the lab. So what we have found in the lab is that when you have these algae growing in beakers like this, we grow them in beakers and provide them with nutrients and light. What we see is that if you add glucose in them, they turn orange. And not only do they turn orange, but they also start producing tags, which has been stained in green over here. So what you're seeing in green are the tags or the lipids or the oil that is being produced by algae. And the red that you see is the chlorophyll or the chloroplast that the algae have, which is photosynthetic in nature. Now, interestingly, when you look at the ultrastructure, as you can see over here, where what we do is we observe cells using transmission electron microscope. And what we see is that there are these structures called chloroplast, which are photosynthetic. But when you add glucose to them, they produce tags and lose the chloroplast. And that is why they turn orange, where they're losing the chloroplast. Food additive known as astaxanthin. So this astaxanthin is also a pigment. It's a carotenoid pigment, which is being produced by these algae. And it is also a great antioxidant, as well as it's a nutraceutical. It's a nutritional or nutrition-rich substance, which is known as astaxanthin. So not only do these algae produce oil, as you can see over here, they produce rich nutraceuticals. So this is what we do in the lab. And apart from that, what we want to do is we want to understand the underlying biological pathways that are involved in producing this oil. And in the lab, we use multiple approaches. One approach that we use is known as a transcriptomic approach. In the transcriptomic approach, what we do is we grow cells in beakers like these, and in one set of cells, we grow them photosynthetically in the presence of light. And in the other set of cells, we grow them in the presence of light and glucose. And what we do is we collect the cells, we break them, we extract something which is known as RNA from them. RNA is what makes protein, which is the functional part of the organism. So we collect and extract the RNA, and we sequence all RNA, and we compare the RNA between the two samples. And with this comparison, what we get is the difference between the expression of the RNA, which would tell us about new pathways that are involved in producing oil. because one set of organisms do not produce while the other set does. So this is how we do a comparison between these two. So we understand which pathways are induced, or rather which pathways are upregulated, as we call it, upon glucose addition. Using this approach, we are able to find key component genes and key component pathways. And an example of this is a pathway, as can be shown over here. So all the yellow boxes that you see are the genes that are only upregulated or induced in the yellow cells, while the blue ones are the ones that are in the green cells. So using this approach, we can build interesting pathways, as you can see over here. And not only can we build these pathways, but we can also understand key genes, because now we can say that, oh, this particular gene is, because it is induced in these cells, we can say that this is important for producing oil. And using this approach, we have been able to identify key pathways, key biological pathways that are important for producing oil. I hope this was clear. If you have any questions, please let me know. Okay, so not only that, we take the biology to at a next level. We want to understand at the molecular level, at the level of proteins, what is going on with the biological pathways and how these pathways are being regulated. So, for example, here I'm showing the central dogma, where you have DNA, which is the genetic code within organisms, which basically is involved in replication, and it forms genomic content, which is being transmitted to the offspring. Now this DNA is converted to RNA and eventually to protein, which is the functional part of the organism. It is the proteins that regulate various pathways. It is the protein that can regulate the oil producing pathway, etc. So what we do in the lab is that using an approach called mutagenesis, where what we do is we induce different genetic modifications, or we damage the genetic the genome of the organism. And what we observe is that whether an organism is able to produce oil or not, suggesting that, for example, a protein, for example, hexokinase, when we isolate a mutant of hexokinase, we see that this protein is very, very important for oil production, because when this protein is absent, the cells are not able to produce oil or turn orange, suggesting that this protein is a key regulator. It's a key master regulator that regulates growth and metabolism. This is how we are able to uncover key proteins that regulate the oil biosynthesis or fatty acid biosynthesis pathway, and using a mutagenesis approach. Now, similarly, we also use a biochemical approach where we use biochemistry. For example, we know that certain proteins are important for growth. And when we inhibit these proteins using biochemical approaches, for example, using an inhibitor, we see that again, this inhibition produces oil and it produces carotenoids or astaxanthin, suggesting that this is also a key player. And this is also a master regulator for production of oil. Now, using this study, we could identify different master regulators that are involved in production of oil. But not only this, we use multiple different approaches for identifying key pathways. And when we understand and identify these key pathways, we also want to improve, or rather engineer organisms to produce, to better utilize CO2 and also produce DAGs or biofuels. And
oh, sorry. So we have a question in the Q&A, and it seems like it might lead well into this next section, where Virginia is asking, are algae more efficient than other biofuel producers, such as corn? So are they already the top dog that you're making even more efficient? Or are they in and of themselves kind of mid-ranked and you're just trying to harness the large mass of algae that we've got?
That's a great question. And so this is a very interesting question, because there is a debate whether algae can be, whether the technology of algae can currently scale up to the level of producing biofuels as is being used with corn. So currently the technology that is available is not at the scale where biofuels can be produced, where they can be efficiently produced and become carbon neutral, while corn is already being produced at an industrial scale and is also being used as a crop, as a food crop. So there are advantages and disadvantages of algae, but currently I would say that in terms of efficiency, because corn is already at a stage where it is being industrially produced and it is being for food, and there is land available, etc. Its comparison with algae wouldn't be that accurate, because the technology that is available to extract and produce and extract biofuels from algae is not at the same stage, if that answers.
Yeah, I think that's great. Thank you. And then I think you also covered this in your answer, but there's just another question saying, is algae or alga currently used at an industrial level to produce oil and carotenoids? And that seems like a no.
Yeah, no, not currently. In fact, I would say that this particular algae that I'm working on right now, which is called Chromochloris ophungensis, is actually one of the top producers of tags or oil that have been suggested by the Department of Energy. However, even this algae is not being used at an industrial scale to produce tags. So not currently, maybe in the next 10 years or 20 years. And I'm quite positively hopeful, because considering that the price per gallon of oil is increasing so much, I mean, it is time to improve the technology to produce algal oil. I feel that is what I feel. Yeah.
Cool. Great. Thank you.
I hope that answers the question. Okay. So another thing I would like to add is that we in the lab are trying to improve ways in which we can improve this oil production, so that the first hurdle in the sense of efficiently producing oil is lost from at least at the research level. So that is what we are trying to do at the research level, in the sense that identify key pathways so that we can engineer and using a targeted approach instead of using just environmental stressors or other aspects. So I hope that that's helpful. So now, so just like I said, we primarily want to identify key pathways that are involved in tag production, so that once the technology is available to scale it up, we know where to target and what to target. So how do we do this? We use multiple approaches. For example, we use something called a transcriptomics approach that I described previously. Similarly, we use a proteomics approach, where instead of looking at RNA, we now look at DNA. We now look at protein. Not only proteins, but we also look at metabolites, as well as we extract various, isolate various mutants using a genetic as well as a biochemical approach. Now, why do we do all this? So that we can identify the key oil-producing pathways and ultimately re-engineer algal cells to produce better biofuels. Now, using this approach, we have had quite a bit of success in the lab. So for example, we have been able to identify 2 key pathways for producing tags. One is known as the de novo pathway, and the other is known as the membrane remodeling pathway. And what we see is that what this means, what does the de novo biosynthesis pathway mean? It means that these algal cells are able to produce oil from if you provide any external carbon source like glucose, and they're able to upregulate various proteins that can produce oil from in a de novo manner, as I describe over here. For example, if you have glucose and you provide them to algae, they can incorporate this externally provided sugar into tags or biofuels. And on the other hand, we have also been able to understand the membrane remodeling pathway, where already existing membranes or already existing lipids within the organism can be engineered to produce tags. Now, the potential of understanding these pathways is great. Once we know the components of these pathways, we are able to target specific proteins and upregulate or downregulate them to accumulate large quantities of tag. So this is the kind of research we do in the lab, and this is all I have for the talk today. I thank my team, which is called the Switch team, and the Neogi lab where I'm from. Laboratory work requires a lot of collaboration, and I'm thankful that I'm at Berkeley, where I can collaborate with a lot of people and do some amazing, exciting science, which we can use to combat climate change. And these are our funding resources. Thank you.
Great. Thank you so much, Shivani. We're happy to take any questions as they roll in the chat and Q&A. But I might start off, and I'm sorry if you already addressed this, but while it looks like algae can do both carbon capturing and fuel production, will those always be 2 separate branches of harnessing algae, or maybe I was missing the crossover? Is it believed that you can do both things?
Absolutely.
Within one.
They can, so they can do both. So they
because you're capturing the CO2 to convert to fats. Okay, sorry.
Absolutely.
I missed that. Yeah.
No, you're fine. But absolutely, they harness the available CO2 and convert it into oil. And these are so what we're trying to do is to identify underlying mechanisms by which they do that. But that is the reason why they are potentially carbon neutral, in the sense that whatever CO2 that is available in the atmosphere is something that they will be utilizing to convert into carbon-rich oils. So that is the reason why they are potentially carbon neutral.
Cool. And is there any predicted timeline for if this kind of process worked? Like, how long would it take to scale this up industrially to actually replace fossil fuels or other non-replenishable fuel sources?
That is a very challenging question, but I'm really excited because so currently there are a lot of places where large-scale algal production is going on for production of biofuels, especially efforts by the Department of Energy and a lot of universities that are putting up. And even in Berkeley, there are a few labs that are trying to do field studies to produce large amounts of produce algae at such a large scale that they can produce biofuels. So in terms of timeline, I would say 2 decades, one or two decades would be something that would, definitely bring algal oil to the market. And in fact, I just want to add that currently, algal oil is being used as a food source in multiple places, and algal biomass is actually used as fish feed and in multiple other places. So things are moving at a pace where you should see that it'll become more cost efficient to use algae as sources of food, as sources of oil, etc., than using, traditional fossil fuel-based sources. So exciting times, I would say.
Cool. Yeah. Looking forward to it. So we do have a question in the Q&A asking, is this alga both simultaneously producing oil and carotenoids and growing well after glucose treatment? So maybe also like, does this production end up kind of killing the algae that's producing it?
No, in fact, they grow really, really well. So when you provide them with glucose in the light, they take up glucose as well as fix the carbon that is available in the light, the proportions of which we are not sure, as in how much glucose and how much CO2 from the atmosphere, but they do require sunlight, because in the dark, they're not able to divide or rather grow as much as they do in the light and glucose condition. So yes, they take up glucose in the presence of sunlight, and they produce carotenoids as well as tags and grow. So there is a huge amount of growth burst that you see. So they also produce large amounts of biomass. So yeah, it's a magical thing. It's a very interesting thing, yeah. The pathways of which we want to understand fundamentally, but yeah.
And does it seem, I mean, obviously they flow into one another, so as much as any biological pathway would be split, does it seem like you have kind of two sides of that coin, where you're both trying to like find the machinery to capture more carbon and to produce more oils? Or does it seem like there might be kind of smaller set of machinery that does both?
Yeah, so this is where we believe our work will really, really help, because the way if we are able to completely distinguish between the carbon dioxide fixing part versus the glucose utilization pathway, then we can probably engineer both in the same organism, and maybe even make it independent of glucose, and just efficiently utilize the carbon dioxide. So this is where I feel like our work will be the most useful. So yeah.
Gotcha. And Sarah's asking, I believe in regards to climate change or just general human activity, are we damaging our environment currently, especially our oceans, enough to harm algal populations?
Yes, yes would be the short answer, but what the research currently is showing is that because of the damage to the environment and because of the heating up of the oceans, a lot of coral species that are present in the oceans are being bleached and are being damaged and are dying. And these corals actually symbiotically live with algae. And without these corals, the symbiotic association of algae is lost. And many of these algal species which symbiotically live with these corals are lost. And it's basically, although high CO2 does boost algal growth to some extent, the diversity per se is significantly being affected. So, yeah.
All right. Well, then hopefully we can convert quickly and save our algal stores. Yeah. And then currently our last question, but we're obviously still open. is whether algae is amenable to genetic transformation or editing.
Yeah, so this alga is something we are currently actively working towards its genetic transformation or editing, so that once we identify these pathways, we can rewire them, etc. But as of today, I would say that the efforts are still going on in the lab. So, yeah.
But currently it's a question of finding the machinery and not how to edit it, or is it also an editing, like how to edit issue as well?
Yeah, so one of my colleagues is working on genetically transforming these algae. So both things are happening parallelly, I would say. Yeah.
Interesting. We'll give a second for any last questions to pop up. But it sounds like we're in for a whole bunch of new discoveries in our lifetime. Algal fuel, life on other planets, all sorts of new knowledge that luckily we're alive to see. So I don't know, this past hour and a half has got me pretty excited about that. Wouldn't want to live in any other time.
Thank you so much for your presentations. These have been just excellent talks, and I'm so glad our listeners could tune in this evening. So I just wanted to remind everyone that tonight's talk is being recorded. Check out Popping the Science Bubble YouTube page in order to watch previous talks. This event happens every third Tuesday of the month. So please come back at the same time, same station for more science talks. All right.
Thanks.
Thank you very much, Kelsey. Thanks, Guy and Shivani.
All right. Thank you. Logging out now.
Great night.